Hello everyone, Chief Symbiont here. Today I want to share with you a demo video from Docent, our AI-based learning platform. Here is how Docent works. Upload a research paper. Our intelligent agent learning partner Sage reads it, builds a plan, and streams a narrated slide deck with custom diagrams as well as the visuals from the paper. You can ask further questions to Sage regarding the slides, the paper, and related topics. Then it quizzes you on what you just learned, finds your weak spots, and reteaches them. That's the symbiotic loop. AI creates and verifies content based on human interaction, then teaches and tests you. You both get smarter. Docent is open source and runs in your browser. Now you will hear from Sage, delivering a journal club talk based on an interesting topic.
Welcome everyone to today's Journal Club. We'll be critically evaluating a recent Nature paper by Xu and colleagues from UC Berkeley and collaborators. The authors build a leaky integrate and fire model of the entire adult Drosophila brain, all 127,400 neurons, using only connectome-derived synaptic weights and predicted neurotransmitter identities, and test whether this remarkably simple model can predict sensory motor circuit function. Let's dig in. Here's our roadmap for today. We'll start by framing the paper's central claim, walk through the model design and key results across two sensory motor systems, and then spend the final portion on critical analysis, what works well, what doesn't, and what questions remain for discussion. Let's frame the central claim. The authors argue that a remarkably simple model, a leaky integrate and fire network with just one free parameter, built on the flywire connectome and neurotransmitter predictions, can predict which neurons participate in sensory motor circuits, which are necessary or sufficient, and how different taste modalities interact. They report 91% accuracy across 164 testable predictions. The two test systems, feeding initiation and antennal grooming, were chosen because both have well-defined sensory inputs and motor outputs contained within the EM volume. A key question for our discussion: is this accuracy impressive, or does it reflect the relative simplicity of the circuits tested? Here is the model architecture and main sugar circuit results, all from Figure 1. On the left, you can see the LIF schematic. When a neuron spikes, it depolarizes downstream neurons in proportion to connectome-derived synaptic weights. Critically, all biophysical parameters are taken from prior Drosophila literature, except for WSyn, the single free parameter controlling synaptic weight magnitude. It was calibrated so that sugar GRN activation at 100 Hz produces roughly 80% of maximal MN9 firing. Note the key assumptions listed. Zero basal firing means the model cannot capture disinhibition. Inhibiting an already silent neuron does nothing. Glutamate is treated as uniformly inhibitory, which the authors acknowledge is a simplification. There's no neuromodulation, no gap junctions, no morphological detail. Ask the group, given these constraints, what kinds of circuits would you expect this model to fail on? The authors use two complementary in silico strategies. First, they activate sugar GRNs and find which downstream neurons fire, the activation screen. Second, they activate sugar GRNs while silencing individual neurons one at a time and measure the drop in MN9 firing, the necessity screen. The model correctly identifies all 10 previously known sugar-responsive cell types, and 8 of 10 are correctly predicted to be sufficient for MN9 activation. The shuffled connectivity control is important. Only 1 of 100 shuffled connectomes activates MN9, showing the predictions depend on actual wiring. However, note the failure cases. Phantom is predicted as inhibitory, but experimentally activates MN9, likely through disinhibition the model cannot capture. A good question here: is the 0 Hz baseline a fatal flaw or a reasonable simplification? This is arguably the strongest validation in the paper. Rather than testing predictions on neurons already known to be involved in feeding, the authors perform an unbiased screen of 106 SEZ cell types using the split Galfor collection. At 50 Hz computational activation, 11 cell types are predicted to activate MN9. 10 of those 11 actually cause rostrum extension when optogenetically activated, a 91% true positive rate.
Of the 95 predicted negatives, only 4 show non-zero rostrum extension, giving a 96% true negative rate. The confusion matrix in panel C summarizes this nicely. However, ask the group to consider, this screen is dominated by negative predictions. 95 of 106 are predicted not to activate MN9. The high accuracy is partly driven by this class imbalance. The 84% accuracy figure excluding this screen tells a more conservative story. Also note that failures concentrate on inhibitory neurons like phantom and tentacular and neuropeptidargic usnea, systematic blind spots of the model. This slide covers the most novel biological finding. IR94E neurons are aversive. Previously, these neurons, which respond to low salt and male genitals, were suggested to mediate attraction. The model predicts they inhibit MN9, similar to bitter but weaker. Panels B and C show the heat maps. Bitter activation eliminates MN9 firing even at high sugar concentrations, while IR94E activation reduces but does not eliminate MN9 at high sugar. The authors validate this quantitative distinction beautifully in panels D and E. Bitter optogenetic activation eliminates PER even to 1M sucrose, but IR94E activation fails to inhibit PER to 1M sucrose. P equals 0.099, not significant. This is a case where the model generated a genuinely novel, falsifiable prediction that was confirmed experimentally. Worth discussing. How confident can we be about a mechanism predicted by a model that ignores neuromodulation, given that taste processing is heavily modulated by hunger state? The model reveals that sugar and water pathways converge extensively at central neurons. At stimulation frequencies producing 40 Hz MN9 firing, 250 neurons are shared between the two pathways out of roughly 380 to 390 total. Appetitive pathways share circuitry. Aversive pathways like bitter and IR94E are largely segregated, with only 2 neurons shared between sugar and bitter. Panel D shows experimental validation. 5 of 6 predicted required water neurons are confirmed by GTAC-ACR1 silencing. The model also predicts synergistic interaction. Co-activating sugar and water GRNs enhances MN9 firing beyond either alone. The authors then show in panel F that silencing sugar GRNs actually reduces proboscis extension to water, which is a novel cross-modal dependence. However, they note a confound. Water-sensing neurons also respond to 50 millimolar sucrose. A good discussion point. Does this extensive overlap suggest a compact appetitive acceptance circuit, or is it an artifact of the model's simplicity? To test generalizability beyond taste, the authors apply the same model to the antennal grooming circuit, a completely separate sensory motor system. Starting from 147 Johnston's organ neurons and asking which descending neurons are activated, the model correctly identifies all 4 known critical nodes. The most interesting result is the Johns subtype analysis. JOCE and JOF both synapse directly onto ABN1, 103 and 78 synapses respectively. Yet the model predicts JOCE will robustly activate ABN1, while JOF will not. Panel H confirms this with calcium imaging. The authors identify 3 putative inhibitory neurons postsynaptic to JOF that may explain this counterintuitive result, a prediction that remains to be tested. This is a compelling demonstration that synaptic count alone doesn't determine activation, and the model captures this nuance through inhibitory wiring.
Let's critically evaluate this work. On the strength side, the model is impressively parsimonious, 1 free parameter for the whole brain. It generates falsifiable predictions, and the authors validate many of them experimentally. The robustness analysis is thorough. Varying WSIN by 30% or the inhibitory to excitatory ratio by 50% only drops accuracy modestly. On the limitation side, the 0 Hz baseline is perhaps the most consequential. Disinhibition, where silencing an inhibitory neuron releases a downstream neuron from tonic inhibition, is a fundamental neural computation that this model cannot capture by design. The phantom failure case illustrates this directly. Phantom inhibits scapula, which inhibits roundup, so phantom should activate MN9 via disinhibition, but the model predicts it as purely inhibitory. Usnea case reveals another blind spot, neuropeptidergic signaling. The authors admirably tested this with Amentalata RNAi, confirming usnea likely acts via neuropeptides. Ask the group. The C. elegans work by Randy et al. Showed that calcium imaging activity propagation is not well predicted by connectivity alone. How do we reconcile that with the high accuracy reported here?
Let's unpack the accuracy claim carefully. The headline figure of 91% across 164 predictions is heavily influenced by the SEZ split GAL4 screen, where 95 of 106 cell types are predicted not to activate MN9, and most of those are indeed true negatives. Excluding this screen, accuracy drops to 84%. That's still good, but it's a different conversation. The robustness tests are reassuring. Wissin plus or minus 30% and inhibitory to excitatory ratio plus or minus 50% cause only modest accuracy drops. However, the glutamate sign test is revealing. If glutamate is treated as excitatory instead of inhibitory, the model loses its ability to predict bitter and IR94E as aversive, and the false positive rate jumps from 1% to 16%. This means the novel IR94E finding depends critically on the glutamate is inhibitory assumption. A broader question. The two circuits tested, feeding initiation and antennal grooming, are both relatively compact feed-forward sensory motor circuits. Would this model work on circuits with recurrence, sustained activity, or heavy neuromodulation? The C. elegans data from Randy et al. Suggest the answer may be no.
Let's wrap up with some open questions for the group. First, consider where this model sits on the spectrum of connectome modeling approaches. Graph traversal models use 0 parameters but only measure path length. This LIF model uses 1 parameter and produces spiking rates. Connectome-constrained deep networks, like Lapalainen et al., use 734 parameters but require task training. Each makes different trade-offs between biological plausibility, interpretability, and predictive power. I'd like to pose 4 discussion questions. First, both test circuits here are relatively feed-forward, sensory input to motor output. Would this model work on circuits that require sustained activity, like the ring attractor for heading direction or memory circuits in the mushroom body? Second, the author's own prior work showed that hunger state dramatically modulates sugar responsiveness. A state-blind model can't capture this. Third, should we view this primarily as a hypothesis generator, a tool to identify candidate neurons for experimental testing, or as genuine evidence that connectivity alone captures circuit function? And fourth, what circuit would you choose as the ideal stress test? Courtship involves extensive neuromodulation, sleep-wake involves tonic firing, and navigation involves recurrent dynamics. These would be informative tests of the model's boundary conditions.
Here are the key references cited throughout this presentation. The primary paper under discussion is Xu et al. 2024 in Nature. Several supporting references include the Flywire connectome papers, the neurotransmitter prediction dataset, prior work on the feeding circuit, and the C. elegans comparison study that provides important context for the model's limitations. Thank you for your attention. To summarize our critical assessment, the paper demonstrates that connectivity and neurotransmitter identity alone can go surprisingly far in predicting circuit composition and function into well-defined sensory motor systems. The 91% accuracy headline is inflated by class imbalance, but even at 84%, this is a strong result for a one-parameter model. The key limitations, inability to model disinhibition, neuromodulation, and neuropeptidargic signaling, are clearly acknowledged by the authors and define the model's applicability boundaries. I'd argue the model's greatest contribution is as a hypothesis generator. It can identify candidate neurons and circuit motifs that experimentalists can then test. Whether connectivity alone truly explains circuit function remains an open and fascinating question. Let's open it up for discussion.
Check out the GitHub repo below and send us your comments. We are looking forward to seeing what you will build based on Docent. Follow us at Cafe Cybernetics for more.